the states as well as union territories. Under this initiative, Envy Center at Indian Institute of Science conduct series of webinars by through the various experts. So today we have with us Subhomita Ghosh Roy. So she is a lecturer from Northern Kentucky University. So she will be sharing her views on effect of land use changes on ecosystem, the indicators. So she did her PhD from University of Wisconsin, Milwaukee. So her research interest includes wetland ecology, microbial ecology, water quality, water treatment, sustainable development, stream ecology, and conservation. She was also a visiting instructor at Marquette University from 2020 to 2021. So she has been a recipient of a Graduate School Travel Award issued by Graduate School University of Wisconsin in the year 2018 August. So she's also recipient of Richard Haumiller Graduate Award issued by the Department of Biological Science in the year 2015. She is also recipient of Clifford Motier Award issued by Department of Biological Science, University of Wisconsin. So she has published six research paper in peer-reviewed international journals and also many reports. She's carrying out various research projects related to the wetland ecology and also the water treatment. So with this brief introduction, I invite Shubhomita Ghosh Roy, madam, to NV's webinar series. So over to you, madam.
Thank you, Bharat, for the lovely introduction. And thank you for inviting me into this program. So, I would today talk to you about effect of land use changes on ecosystems and the indicators. So, when we say that when we use this term land use, what do we mean by that? Is like how we are using the land that is available around us. So, land use is something that is not natural. What we have naturally on earth, it's land cover. Now how we change it accordingly, according to our requirement, human requirement, that is land use. So for the brief outline, so initially I'll talk to you about like how this as per the introduction, like I'll talk to you about how the land use changes can cause human risk, and how this risk is really propagated, from where it starts and where it goes, how it can be assessed, and what are the roles of various bioindicators in this matter. Then I'll talk to you about my first study, what is the watershed I studied, how I studied it, what are the bioindicators. Then I'll go into the second study, which was done in the same watershed, but that was an extensive field study. So I'll talk to you about that and what kind of indicators I found in that study. Then the third study, which was not so much field intensive, but it was about calibration of the previous study. So I'll talk to you about that and some specific indicators that I found there. I'll sum up all these studies, and then I'll talk to you about another ecosystem, which is a pilot study that I did in India that was on soil ecosystem. And then I'll talk to you about the current study that I'm working in, which is Ohio River Watershed here in Kentucky and Ohio, and then the future direction and and final conclusion. So to begin with, how risk is seen by us and how risk originates and how risk is measured. So root risks come from, or ecological risk, when I say about risk, it is basically the risk is given by us to the ecosystem, and that is ecological risk. So where it come from, it can come from a regional scale, or it can come from a local scale. For example, there is a lot of atmospheric deposition, that's a regional stress factor that can result into ecological risk. It can be wetland damage, it can be a lot of urban land use, it can be forest damage, all that done in regional scale. Or it can be simply at a local scale, for example, like a lot of construction going on in some area, and that will land up in a lot of pollutants, a lot of traffics, fertilizers being used a lot, impervious surfaces, increased land uses. So that those are local scale stresses, which will lead up into which will lead the environment to be exposed to the stress factors, and that will mean that the environmental quality will deteriorate. So there will be changes in physical habitat, the water quality would deteriorate, the sediment quality would deteriorate. And And to know about this exposure, to know about this risk, we have to go and measure some endpoints. For example, we can measure population fragmentation, population or species that was living in that ecosystem, or what effect was created in the aquatic species. And how we measure it, we choose some specific endpoints of that. For example, how was the aquatic benthic organisms were affected by this, how the diatoms were affected, how the fish growth was affected. Those would be our the ecological endpoints. And if that happens, why we measure these endpoints? Because these are the endpoints that we can relate to really to human health. So therefore, that's how risk is generated. This from the root factors are there, and how it is layered, and then it lands up in the final endpoint. So in US, when it comes to risk assessment, and probably this is followed globally also, so a big stool that was used in even till in last century was the three-leg stool. It was proposed by US EPA, which is Environmental Protection Agency in US. So they proposed three different layers. First was, they said that if we want to measure an ecological risk, then we should measure the water qualities, we should measure the effluent toxicity testing, or the we should do some biological assays. But soon that was seen not being very much like enough. So there was another approach that was taken, and this is kind of a widely accepted, globally accepted way of assessing risk. So in this case, we would measure chemical risk, ecotoxicological risk, like how the toxicity is being generated into the ecosystem itself, and then what is the ecosystem status, what is the health of the ecosystem. That's called the ecological risk. But these these researchers kind of showed us that this is fine, but we still need to incorporate more of biological response signatures, like something that is that can tell us more about the biological organisms or biology in that ecosystem. So in that regard, then said, if we have to have more effective monitoring strategies, then we should have bioindicators coming from different trophic levels. So, what are bioindicators? Bioindicators are some form of organism which are capable enough to show the ecosystem risk status, like the ecosystem contamination, the pollution status. They can be tolerant to that risk compared to the other organisms, or can be too sensitive. So in either way, they would kind of show us the different aspect of this risk assessment. So my entire research has been based on this idea of bioindicators and how it can assess, and coming from different, particularly coming from different trophic levels. So that's the basic research model I use. So it starts from, if there is population growth, then there will be environmental challenges, which means that the natural land cover being converted into artificial land use, and that will generate more storm water. Why? Because land use isn't something very artificial, and that will increase the impervious surfaces. So, if it's a human used land, then there will be pollutants. And those pollutants, for example, if I create a parking lot, or I create a driveway in front of my house, that's not a natural land use, that's an artificial land use. And if there is a pollutant generated, then that will not land, that won't be able to percolate through the soil ecosystem anymore. So it will go through the rainwater, the storm water, and that will land up in the nearby, those pollutants that is being generated there, they will land up in the nearby surface ecosystems. For example, wetland. So wetland is one of the major ecosystem that I have studied. In that way, the wetland water and the sediment, they both can get contaminated. And in order to measure that, I have used again two different types of bioindicators, coming from two different trophic levels. Firstly, ecotoxicological, which are plants mainly, and then some biological response signatures, which are the native bacterial species that is living in that ecosystem. So the first ecosystem that I worked, that was Pike River watershed, Racine in Wisconsin, and this is the entire watershed. How it looks like, it's a heavily urbanized, highly populated watershed, and it's just beside Lake Michigan. So that's how this watershed looks like. So the reason I chose this watershed, because this, like I said, it is a rapidly urbanizing, lots of land use changes, and barely any undeveloped land being left here. So like you can see, this is the stream, and because of this urbanization, this watershed also went through a lot of problems, because it was generating a ton of floods in that area, and then damage of property and agricultural lands, because there was not enough stormwater retention areas. So this river went through a major restoration project. All these, like you can see in this 2015 picture, and probably some in 2002, all these stormwater retention ponds or wetlands were created. At the same time, it also underwent a lot of urbanization. Like you can see in this insets, like over here there was almost nothing, and then it started developing farmlands, and these are residential areas. Even more here, it was a totally undeveloped land in 2002, and within 13 years, lot of parking lots, commercial spaces, malls, parking park trails like came into there. So this is a rapidly changing watershed, and so I chose this so that I can catch the impact of these urbanizations. Mainly, currently, Pike River watershed has these types of land uses. Industrial, there is a big factory of AC Johnson's there. There are people who live there. So residential, commercial means lots of shopping malls and parking lots. Some are undeveloped, and a major part is also agricultural. So this distribution actually, or this graph shows like how there are six wetland sites that I chose, which I will come in a second, that I worked with, and how the land use is like different in each among them. So some of have like high amount of agricultural uses, some of them have high amount of industrial and commercial usage. So that's the entire study site. So this is Lake Michigan, and Wisconsin is is one of the Midwestern state, and this particular watershed is in the southeastern part of Wisconsin, and that's the entire watersheds. And the study sites that I worked in, which was around the northern part of this, and these are the six wetland sites that I studied, and some of them are natural, but most of them were created during the restoration process in order to control flood in that area. So after these sites were chosen, then the next thing that I did, like how there was no reference point in these wetland sites. Like we did not have any idea that how much kind of pollutant are being generated in this watershed. So but I could calculate the area using data from SWRPAC, which I like just showed you here. Then SWRPAC is Southeastern Wisconsin Regional Planning Commission. So using their data, I was able to know the percentage of land use and area. So use that data. Then I did a literature review which has this similar kind of land use types, and they are pretty similar in terms of they're not very far away from also this location, but not exactly this location. So I found some reference points there, so I extrapolated their pollutant loads and extrapolated that on my wetlands, on these wetland sites, and calculated some possible predicted loads just to give us like a reference idea. And the pollutants that we chose was like nitrate, phosphate, basically nitrogen or nutrients, and then bunch of metals, cadmium, copper, lead, and zinc. So all that gave us a kind of a reference point that what could be the possible predicted load in a year possibly going on in this wetland sites. So therefore, then in my first study, the first question that I addressed, like, do the plant bioindicators, and I will just come into a second, like what are these plant indicators, do they correlate with this predicted loads that I talked about, predicted loads of nutrients and metals in this particular wetland watershed. So I sampled across different seasons across these wetlands. I mainly used core sampler and Ekman rate, so I had to get the sediments out of these ponds. And this is the ecotoxicological test that using and I use these plants as the bioindicator. So this test is called a phytotox kit. This is a standard test developed by Microbiotest Belgium. It comes with three bioindicator plants that's being already tested. So sorghum, lepidium, sinapis. And what we have to do, it also comes with this kind of test plate. So we put around 300 grams of sediments at one of the compartments, saturated water, cover it with filter paper and add like 10 seeds on that, cover it and put it in darkness for 72 hours at 25 degrees Celsius. After 3 days, we'll take it out and then we'll measure the stem and root lengths of these plants. And we do the same thing for the test sediment, the sediment that I'm interested in working. And also there are some control sediments which are like washed sands. So using that, and then I will, using those two data, control and the test sediment, then I will calculate the possible proportion of root and stem growth inhibition that might have happened in the test sediments. So that those are the three bioindicators, plant bioindicators that I used. So the first results, what it showed me, again, this is not like the real measurement of the pollutants, but this was a reference point for us. So we just wanted to know how these pollutants really react to these bioindicators in this particular watershed. So what we got to know, firstly, there is a difference in response among the plants. For example, this plant sorghum, which is a mustard plant, a species of mustard. So they have highest amount of response, and this would be pretty consistent throughout the study. And other than that, with increase of nitrogen loads, particularly nitrate nitrite, in particularly in stem growth inhibition, there was a drop in inhibition. And when we say there is a drop in inhibition, it means that the growth was really facilitated. So it helped in growing. Although we did not get much results in the root, the phosphate results were not statistically significant, but in this case, phosphate was not facilitating their growth, but growth inhibition was increased. So phosphate was not really favoring the stem growth inhibition. So with these are a little bit contradictory, and there are some contradictions in these results, but also not surprising because sometimes phosphate toxicity can occur, and nutrients, if it is overloaded, then it is not good for organisms either. So that was the first preliminary result we got. We did the same thing with metals also. We again saw some interesting more response from sorghum species, and lead was again gave us some very significant, statistically significant results. With increase of lead, there was drop in inhibition, and this was an interesting finding throughout. This is going to, lead had a way different defined story in this ecosystem. So it also means that there is a possibility when we actually go out and sample sediments from these ecosystem, we might see that lead is really getting not like it is by, it is probably accumulated in these plant tissues. There was this possibility. So then that's what we probably would find out in the field study. So with that, we concluded in our first study that there was some result, some significant results, for example, like in lead, but mostly the significant effect of nutrient loadings, like particularly nitrate nitrate, was found on these bioindicator plants, and there was a pretty much correlation of these pollutants with the nutrient with the bioindicators. And that's what we wanted to know initially, like if there is at all a correlation or not. Do they tell us any story? Is there any response that we can get there? So the next question was that what would happen if we go out in the ecosystem and measure these pollutants, and if we can use any specific bioindicators that is already in the ecosystem, means like some organism that is already living in the ecosystem, if we can use that as a bioindicator. So in the second study, the first research question was that do these plant bioindicators and the taxonomical diversity, or the bacterial taxonomical diversity, or the bacteria that is living in the ecosystem, do they correlate with these pollutant related stresses like nutrients and metals in this watershed? And there is a second question also which I'll discuss later. So for now, we would want to see if this bioindicators really correlate with pollutants that is directly being measured, not predicted anymore. So we used the same samples, and we got some more water samples in this case because we wanted to measure nutrients, some from the same wetland sites. So the bioindicators, the first one was the same one that we were using before, the plant bioindicators. And then the second one was this in situ bacterial bioindicators. So for that, we extracted DNA from sediments from these wetland sediments. We did a library preparation and sequence of V3, V4 region of the 16SR RNA gene using Illumina next generation sequencing. Although I did not do the sequencing myself, I did like the DNA extraction, PCR, and we did not have the facility to do all the sequencing steps. So we had to send it over to UW Madison to do the sequencing. Then I received the sequences electronically, and then I ran the bioinformatics. We use a particular software called Mother, which kind of gave us the entire taxonomical profile of what could be the possible bacterial genera that's living in this ecosystem. So with that, I calculated two diversity indices, Simpson and Shannon, which is a very standard diversity indices used across all many ecological studies. In terms of the stress factors, we measure two things. Firstly, nitrate and phosphate, which is the nutrients in water, with this given detection ranges, and then metals in the wetland sediments. And in this case, we used XRF technology, which is Bruker Tracer 3.5 spectrophotometer. And they were calibrated. All this instrument was calibrated using standard reference material before we could even proceed. So the first results showed us, like, now we're talking about some measurements that we have done in the ecosystem. So the nitrate results was pretty much what we saw in the inhibition, I mean, in the predicted results as well. So with increasing nitrate load, there is drop in inhibition. So nitrate is still facilitating the growth. We still see sorghum being the most responsive species, but in this case, in the predicted results, we did not see much response from the root inhibition. But in this case, we do see that with increasing nitrate and phosphate, there is increasing inhibition. So in this case, with the same stress factor, nitrate, this plants are behaving in a different way when it comes to root growth and stem growth. So the root growth is not really favored by nitrate, but stem growth is. So that means that these nutrients are working or interacting differently with this plant parts even. So then these are what the metals that we found in the sediments: Ag, Zn, zinc, arsenic, cadmium, nickel, lead, and mercury. And then we ran a principal component analysis on them. So principal component analysis is a statistical tool that kind of summarizes a large amount of information, large amount of content, and group them into smaller data sets, and provided that they are closely related to each other. So in terms of concentration, all this bunch of metals, they were kind of similar in their concentration. So they were grouped as one group, and they were put under metal factor 1 or component 1, but in the positive axis. So they were kind of present in much significant amount. And the other two metals, lead and zinc, their concentration was slightly different than these ones. So they were at a different axis, so they were under the metal factor 2. But lead was actually present more, so that's why it is in the positive axis. But mercury was actually present very less, so that's why its concentration is related to lead, but not that much also. So that's why it's in the negative axis. So which means we will see mercury at the negative axis, which simply means that mercury was present there but at low concentration. So when we analyze these results using these plant indicators, we again see a more response from the sorghum. But I would like to draw your attention in this figure, where we see like how the results are showing a pretty similar story what we have seen before. So before, we did see that with the increase in the predicted results, with increase in lead, there is drop in inhibition. And there we see the same thing, because if you remember here, in the positive axis of metal factor 2, lead is present. So with increased amount of lead, there is drop in inhibition. But mercury, even though it was present in smaller concentration because it is in the negative part, still it is kind of inhibiting the root growth. So when mercury is there, inhibition is high. So this was one of the most interesting finding. So therefore, definitely these plant species, and particularly sorghum, were related highly, like kind of have a big significant correlation with these metals, particularly mercury and lead and phosphate. So then we wanted to know what is happening in the bacterial indicators. So first we plotted the two diversities against like the nutrient concentration. So with, for example, with increasing nitrate concentration, we see there is a drop in Simpson diversity. So too much nitrate is not really favoring the diversity in this case. But with phosphate concentration, we also see similar, and these two results were very much statistically significant. So therefore we see that too much of these nutrients are not really favoring the diversity. We did the same analysis for the metals also. Again, we see a pretty similar story what we saw in the plant bioindicators. So lead being present here, the diversity is high, and diversity is high means that lead is really not impacting this bacterial uh, community negatively. So there must be something special about this community that it's still about to thrive in lead, because lead is a poison, but that still the plants are even thriving and even there's diversity. And we will kind of figure out why later when we little bit go further, that why is this happening. And then even the mercury was not like that. So mercury is still in this negative axis and the diversity is pretty low in here. And compared to the other metals, which is zinc, arsenic, and cadmium, nickel, these metals are not really favoring the diversity, and and we saw some significant results that diversity being like getting reduced. But then the question is that there is something interesting about lead here. So we did found some significant correlation in this case also for the pollutants and the bacterial indicators as well. Now, so we did answer our first question. We found some significant correlations. Now, the next thing we wanted to know, that is there any specific kind of combination, special type of bacterial taxa that could be possibly a predictive indicator of pollutant loadings? What is going on? Particularly, we were interested about this lead thing. Like, why is it like that? So we analyzed the taxonomical data. This is the major kind of phyla that we found there. And this is pretty typical in a soil or sediment ecosystem. These were the major genera. This is just some examples of the genera. There are many more. I could not fit it in one slide. So this is kind of will show you the idea of the how much diversity we had. This is just the 6 samples from one particular season, all the genera that we detected in 2017 summer. And then, so before we could proceed further, so because we got the taxonomical profiles already, so we did a literature review again. And why was this review done? Just to see that the genera that we have detected, is there already other literatures, other scientists have already, whether they have detected some special type of bacteria that could also justify our results. For example, are there already some detected bacteria as phosphorus solubilizing or lead resistant or mercury resistant? So these were the different examples that we found. So we use this data, and then because we already have the taxonomical profile, and then we were able to like categorize what taxonomical profile we found into 6 different categories when it comes to our results. So the first category, so some bacteria were PSMs, which is phosphorus solubilizing, and lead resistant and mercury resistant, which is Bacillus and Pseudomonas. Then some were PSM and lead resistant, some were just PSMs like Thiobacillus and Rhizobium. Some were simply mercury resistant, some were both mercury and lead resistant. But the most interesting bacteria was species we were interested is this. There were lot of lead resistant genera being said, like coming from Staphylococcus, Escherichia, Nitrospira, Arthrobacter, Cuprividus, and many more. This is just again some examples. So therefore we could justify the why we were seeing increase in diversity or with lead or like drop in inhibition, because the concentration in lead were present. So there are a quite significant amount of lead resistant bacteria. So even though this wetland sites have lead in there, and which is not surprising because it's close to industries and commercial lots, and they often generate this kind of metal heavy metal loaded pollutants. So even though there is lead, but there is some special type of bacterial indicators, so which is kind of justifying why they are still able to maintain that diversity. So therefore Definitely these bioindicators were able to detect stress from the ecosystem. We found some specific types also, and what we found out that lead has a very significant story in this particular ecosystem. Although there are some contradictions to the results. Firstly, even if we are using the same stress factor, but uh, different indicators are responding to that a little bit differently. For example, root and stem can be different, or the different to diversity indices can react differently, which is also not unusual because there are different parameters. So they are kind of showing us, can show us the different stories sometimes. But the most important thing that we were interested in, that what could happen if we increase the stress. And this is a natural ecosystem, so stress sometimes can be really high, stress can be sometimes really low. So we wanted to know, if the stress is really high, then how will these indicators like react to that. So in order to do that, we had to do an in-house in lab study, so that we could calibrate our indicators against high concentration. And we made like wetland microcosms. So that's how these were made. So we got some sediments from these same wetland sites, and we made these microcosms built with the sediments. So again, we chose four wetland sites out of the six, and we collected sediments from there and built microcosms like this. So I'll come back to this slide. So the microcosms would look like, typically used to be like this. So they were like 18 by 22 centimeter bucket. We would fill it up with sediments up to 5 centimeter and fill it up to RO water up to 10 centimeter. And then we would add the different stress factors into it. For example, in the metal treatments, we would add definitely lead, because lead is our concern here, and then copper, because copper has, see, previous studies also kind of showed us that copper also could be a possible another metal of concern. So we used copper in this case, and then nitrate and phosphate for the nutrient treatments. And the way we designed this experiment was like, we built this microcosms, added this nutrients, and then took some data out, like what is the water nutrient being present, what is the water metals. In this case, we also measured metals in the water because we were adding the metals in the water, and then status of metals in the sediments. We did that in day 0, 1, 4, and 7. And at day 0, we also kept some sediments aside for doing our sediment studies, the ecotox and the bacterial studies. And at day 7, we decanted the water, drained off the water, and added new water, again, added new pollutants into that. And this was a cycle. So 7 day was the average retention period. And we collected the sediment samples at day 0, 7, and then at the end of the experiment. So that's how the experiment design was done. And so therefore, the question that we generated for, or we had for the study, like, how will these indicators that we have used before, how will they correlate with this manipulated increased change of these stress factors in an experimental system? And also the second question was, obviously, do we again see some specific assemblage of bacterial taxa? So in order to, we'll talk about first about this first question, like how do the manipulated change and these indicators relate to each other. Firstly, in general, we see a big decrease in these pollutants overall. So there was decrease, for example, in lead. This was lead in water, in copper, in nitrate, as well as in phosphate. These are high here because it was phosphate just being added, and phosphate as a nutrient is typically contained in the sediment but takes slight time to get contained into the sediment after it is being added into the water. So that's why we kind of see a spike in here. But in general, from beginning to the end, there was a drop in the sediment, which is not surprising because these are wetland sediments, and that is one of the major function of wetland sediment, to contain pollutants in them, assimilate pollutants in them. In terms of the metal sediments, there was not huge variation among the metals, but again, lead was kind of interesting here. So we do see from the beginning to the end of the experiment, there is actually increase in lead. In case of water, we see that there is a drop, but in sediments, we see there is increase in lead. So it's very evident that the lead that was given to the water, that was slowly getting assimilated into the sediment. So that was the most significant thing in among all of the other metals. Now, when we analyze the results, what we saw, like very much before, like lead was sort of favoring these diversities of these bacterial indices, we see the same thing here. So even though we have added lead in higher concentrations in these waters, we still see that there is increase in diversity. But that was not the story for copper. There is, because copper, although was kind of affecting the diversity negatively, so there was a clear decrease in diversity. And then we kind of see the similar trend with zinc also, like with increase in zinc there was increase in diversity also, but although we did not add zinc in here, it was naturally present there in the sediments. But this is not surprising because probably zinc was not present at a high concentration and that was not enough to affect the diversity. But again, lead is something that we would focus on. So in terms of the plant indicators, I don't know if you can see here. So this is lead concentration in water. Against this lead concentration in water, we see that increase in inhibition here. So this is something different that we have seen before, because lead was really not affecting the growth inhibitions, or not affecting the growths. So, but here it is. Now that's a interesting find, because we have added a lot more amount of lead in the water. Now that we have increased the lead content, now the plants probably are reacting differently to the lead content, and that's why inhibition is affected more, which is significant in statistically also. So in this study also we did see a significant relationship, but firstly overall we saw a reduction in the pollutant amounts, be it all of the pollutants that we have added: lead, copper, nitrate, and phosphate. With increase in lead, we see an increase in inhibition in case of sorghum, which was different than before. Copper was affecting also the diversities negatively, but lead again was, with increasing in lead, there was increase in diversity again. So this was again our focus point. So we wanted to see in our second question again if there is some specific type of bacteria, a specific type of assemblage of bacteria that is there or not, so that this relationship could be figured out. So, in order to do that, in whatever kind of taxonomical profiles we found in this ecosystem, we categorize them in four distinct categories, and we were able to do that. And we used cluster analysis for doing this, and we did that for all the wetland sites. And this is just one example, the first wetland site. So we did this cluster analysis based on the abundance of these bacterial species in the wetland sites. So more the red means that particular bacteria is more abundant. So based on that, we found four categories. For example, these bacteria like Exujobacterium or Psychobacter, they were intolerant because they were present initially, then they more or less disappeared from the system. Then some were sensitive because they were present and they tried to come back a little bit, but not so much. So they were sort of sensitive. And then the two tolerant categories, which highly tolerant means is totally red, like it was present all over the experiment. Like for example, these species are all highly tolerant, but some were not so much tolerant, little bit less, so they were less tolerant, so they were less red actually. So we were able to do this for all the wetland sites, and this is kind of a snapshot of what we found. So we found like some bacterial species like Exujobacterium, Psychobacter, Chlorobium being intolerant. Some were sensitive like Nitrospirus, Phingomonas, Agrobacterium. Some were less tolerant, and some were highly tolerant. For example, Pseudomonas, Flavobacterium, Ralstonia, Artrobacter, Thiobacillus. Now, interesting thing is these underlined ones are the ones that we also detected in the field experiment. So there are some, again, some highly tolerant bacteria, and among these yellow ones are also lead resistant, and some of them are even copper resistant. But there were again many lead resistant bacteria, which is dominantly because we have analyzed the abundance factor here now also. So we do know that these were present here abundantly in these ecosystems. So therefore the we could justify that why lead was favoring the diversity, because many of these bacterial species were really tolerant to, or they were resistant to lead. So conclusion is that the sediment bacterial indicators all of them were highly correlated with these metal factors. And we again found some predictive indicators very similar to the field study. And this sort of gave us a direction towards the calibration factor. So, to sum up, we did study a riparian wetland ecosystem, and we used plant bioindicators that were grown on the wetland system. Then we also used a bacterial indicator by extracting DNA from the sediment directly. And then we found some correlation among these pollutants and like nitrate, phosphate, and heavy metals and these indicators. And then we also found some significant category specific categories of bacterial bioindicators. But one of the major thing that we were not able to do, that was not exactly in scope of this study, that we were not able to figure out the specific species. And if we could do that in future, that would give us a more robust idea, like what is the specific species, so that we know that what kind of genetic uh, composition is there, that these indicators are doing that they are so much so lead resistant. So that is one thing that is not done in this particular study, and that was not within the scope of the study rather. So the next study that I'm going to talk about, this was done in India in 2019, and this was a very much a pilot study. But the goal of this study was again to assess risk, but at a different ecosystem. It was a soil ecosystem, and it was being contaminated by human caused reasons. For example, there are a part of India's textile dye industry is informal, and they just dump their waste directly into these soil ecosystems. And that was a stress factor over here. And the reason they do that, because they don't have proper access to waste disposal, and which therefore it can definitely contaminate the surface water, groundwater, and definitely the topsoil. So we wanted to assess that risk against some known indicators that we have used before, like these plant indicators. And we wanted to know if they can detect any response or any form of toxicity that the soil is being contaminated. So the method we used was the phytotox kit that we have used before. We used again extra fluorescence technology that we have done before, but in this case, this X-ray XRF was used in field, not in exactly. We were able to bring back the samples in lab, so we had to measure them in field. These were the two locations, location A and location B. This was not very far away from Bangalore, actually. It was around the area of Bangalore. And that's how the sampling locations were done. And both these cases, these were the standard dyes that they use as their in their industry. And what we first see, that what kind of metals were present in their dyes. So these were the metals like including iron, copper, lead, again then rubidium, strontium, zirconium, molybdenum, and even gold. So these were present in these dyes. And we also see presence of these metals in these locations also. For example, in location B, where we found the most interesting results. So in location B, we do see lead, zinc being present, then strontium, zirconium, iron, rubidium, copper, all of that being present. But this does not necessarily mean that the only source of these metals in these soils are from the dyes, because metals are present in soils naturally also. But it is possibly there is some contamination because they are being heavily dumped historically over long time. So this is the results from the location B area. So what we saw here, unlike our previous study, here the root and stem kind of gave us a similar indication. We did the same principal component analysis. So PC1 means principal component 1 and PC2 means principal component 2. And in this case, this bunch of metals were under component 1 and lead zinc was in component 2. So we do see with the increase in principal component 1, there is increase in inhibition. That clearly kind of tells us that the growth of these plants were inhibited in presence of these bunch of metals, which is strontium, zirconium, iron, rubidium, copper, manganese, and all of that in combination. But that was not so much the case for component 2, which is lead and zinc. So, although this was a pilot study and these were some very preliminary results, so possibly these soils or these sites are contaminated with these dyes. But there, and also it kind of tells us these combinations of metals that being dumped there, that is kind of giving us a response from these bioindicators. So which this trend, which obviously needs to be investigated further, that why we do see that kind of trend. So now I'm going to talk about a completely different study that I'm currently doing in. This is in Ohio River watershed. So Ohio River is a huge river in Midwestern US. It kind of flows through 7 states in US, including Kentucky is where I live. And it's 981 miles long, which is 1582 kilometers, and 205,000 square miles is the drainage basin. Its average depth is approximately 24 feet, and it's very high in rich diversity. 30 million people live here, and among 5 million, drinking water is being supplied to 5 million people. But it has a big non-point source pollutions basically. So it mainly coming from urban runoffs. There are a lot of urban developments are were previously there, and they're still developing agricultural activities, and also some abundant mines that used to exist in this area, not anymore. So that is the source of pollution in this river watershed. So this is a study that was done in Ohio River watershed in 2015, that kind of gives us an idea of the types of land use and how the land use have changed. So more the red means more urbanization has increased. So like from 1930 to 70 to 2010, this is the recent most picture. Urbanization has increased, the red component have increased, and it is projected that it will increase even more. So, for example, in typically in this area, there is a critical limit of 10%. Like in a river watershed, urban uh, land use should not increase more than 10%, but it is projected that it will increase very soon. So, urban land use is going to increase, but agricultural land use will decrease. So, this is the impact that I'm interested in, that how that is impacting Ohio River watershed. So the questions, then again, this is a very preliminary study, but the questions I want to investigate here, like what is the status of this ecosystem itself? Like for example, the what does the water quality tell us? What are the how are the water quality characters are changing? And what is the status of some in situ macroinvertebrates or some organism that is already living in this ecosystem? And if what is their status, and if that status can be used as a biological response signature or as a bioindicator itself. So I'm currently being focusing on two sites. One is a Covington site, which is around here. That's the river, and this is the Licking River. So, and this is a tributary of the Ohio River, and this is a heavily urbanized site. And Cincinnati is a very like it's a big city, and across that is Newport, and Covington is this location, and this is actually where I live and work also. But this is a heavily urbanized site. So this is the first site, Covington site, and the next one is Markland site. So this is not technically urbanized, but it has a different kind of human impact. There is a dam in there. There are a ton of dams across the river, and this is one of the major dam, and this is called a Markland Dam, and it's also surrounded by agricultural land uses. So this data is primarily collected from Orsanco. Or Orsanco is basically the River Valley Water Sanitization Commission. So they already have these data historically collected. So I'm for the primary investigation, I'm just using their data for now. So in terms of the water quality characteristics, I'm studying currently like how the pH and temperature is changing. We do not see much of variation among pH among the sites. Dissolved oxygen is also not too bad. It's kind of okay for it's about between 6 to 14 milligram per liter, so it's not that bad. Specific conductance, there is difference among the two sites. So specific conductance, like you can see, is higher in this Markland side compared to the Covington side. But the suspended solids are also sort of high in the conductance compared to the Markland sites. So these two are very specific indicators of water quality and water pollutants being added, though, so which we will actually investigate later. In terms of nitrate is very high in Markland compared to Covington, which is not surprising because nitrate is used in pesticides and fertilizers, and Markland has a lot of agricultural sites in that area, which is not present in Covington. Although the phosphorus content was sort of low in Markland side. In terms of, we detected some metals also. We found like there are some existing metals for like lead, zinc, arsenic, mercury. Although this is needs to be investigated further about these metals. Then we also found some macroinvertebrates, like the types of number and type of macroinvertebrate that is possibly present here. And currently, I was able to retrieve like years from four data from 4 years, 2000, 2001, 19, and 21. But from there we could directly see that there is a drop in the total number as well as so much in the type of invertebrate as well. So that is something interesting, although I have to incorporate some data. I had to retrieve that data from Orsanko and incorporate data in the middle years, like between 2000, 2001 and 2021, and then the story could be more clearer. But even though with this preliminary investigation we do see there's a drop in this number and type, so we sort of investigated the abundance factor, like what are the majorly abundant macroinvertebrates. So these are the percentages. But interestingly, what we see that across all the years, always just 4 years, but across all the years, some species, which is highlighted in red here, these are kind of highly abundant in all of the sites. And these are mainly like some form of mosquitoes, some form of small other types of invertebrates like mites and all that. So these are pretty much consistent across all of the sites. So there is a possibility that in future they can become like possibly a bioindicator. But another interesting thing that we have Dreissena polymorpha, which is an invasive species that is also very abundant, which is a very invasive species in this area. It's zebra mussel actually. So that was kind of disappointing to see that they are also abundant. So therefore, final conclusion and the future direction. So we started from calculating the triad approach of the integrated index, which incorporates chemical risk, toxicological risk, and ecological risk. And the triad approach tells us to see more biological response signatures in terms of bioindicators. And we have done that particularly in Pike River and to some extent in the India study as well. And also, so we did study heavy metals and nutrients, which tells a story about the chemical risk. We did use some plant bioindicators, which tells us about the ecotoxicological risk, and then some bacterial indicators, which is possibly an ecological risk, tells us the story directly out of the ecosystem. But currently, what I'm interested in is more into this ecological risk area. So, if, for example, in this Ohio River study, my focus would be more into like what are the native macroinvertebrates or diatoms or even fishes or even bacteria, could be harmful bacteria also. Like, what are their status? Are they at risk or not? Because that also gives us a story about the ecosystem status and if they can be the bacterial indicator itself. So, with that, I would like to thank all my associates, including my PhD advisor, Dr. Alinger, and my colleagues here at NKU, and my friends and family. And with that, I would like to take your questions.
Thank you, madam. Thank you so much for sharing your vast knowledge. So there are some questions. I will share you the questions. So there is a question from Varun Sharma. Madam, do you think climate change is having any role in pollution and water availability?
Oh, yeah, of course, very much so. So, like we are seeing this infrequent flooding. So, I mean, areas are getting flooded where it's not supposed to get flood. There are tornadoes and storms happening in areas where it's not supposed to have any, opposed to area where it is like really abundant. So, for example, in just few weeks before, we had a huge tornado in Kentucky over here. So, and Kentucky is a like just in middle of the country. We there is not much water bodies or other than Ohio River. So it's not supposed to have this kind of storms, but it was flooded and it was a very significant flood. Millions of people lost their homes. So therefore, that is a huge impact. So we will see this different discrepancies in water availability in different areas. So that's why, like, where it is supposed to have more water, they might run out of water because of these differences in precipitation, and some drier areas might have like a lot more water than they're expected to have. So yes, there will be a huge effect of climate change on water availability, and that is true across the globe, not only in US. It's true across the planet, and as we go further we're going to see it more.
Okay, thank you. Uh, there is a question from Nishchit Kumar Mondal. Madam, did bioindicators vary from watershed to watershed across various locations?
Yes, so if I am definitely to it, because if I am using like bioindicators from the ecosystem itself, then it will vary. For example, in Ohio River, we are expecting that these macroinvertebrates will be the could be the bioindicator. But if I go and study this in India, macroinvertebrate could be an indicator, but it will be some different species, just because it's a different type of ecosystem or different climate basically. Even if I do and study like bacterial bacterial study in here, what I found in Pike River, that will be different in here, because again these are two different places and like vastly away from each other. So yes, it will vary. There will be still some similarity, like some organisms will probably be present universally, but there will be a lot of differences, particularly across different climate zones, different countries, different continents. Yes, there will be a lot of difference. So we
Okay,
have to, when we study bioindicators, we have to take that into account.
okay. So there is a question from Manasvi Kiran. Is there any change in nutrient loading across the study area? If yes, how to address the various loading and their impact?
Well, nutrient loading, yes, it is different. It would be different in across my study area. For example, in some, like, I'm not sure if he's asking or she's asking about this particular site or in general. Did Was that clarified in the question?
No, she's mainly looking at across the study area with respect to the accumulation as well as nutrient load.
Okay, yeah. So across the study area, yes, there would be some different types of different kinds of I mean, nutrient can be loaded differently at this even different wetland sites also. So, for example, let me go back to one of my initial sites, initial slide. So, for example, over here, like you can see like the how the nutrient load or the phosphate load, even the metal load is different among the study sites. So they were different, but if what we did, we had to incorporate that all together in order to study that. So our goal is to see how the nutrients are responding, I mean how the bioindicators are responding to this overall load. But if we want to see across different wetland sites, that is also doable. Like that would make the study more like that would be more like intensive, but we can do that. Like we can actually see in each particular site. In that case, the nutrient loading would be different. There would be difference in if we go and measure, it would be highly different, like very much different across the wetland site. So yes, it would be different, but and that is also can be doable.
Okay. So there is another question from Nishchit Kumar. Is there any study regarding accumulation of these metals, especially from dye industry effluent, into the plant body?
There could be some, yes. I mean, I know there have been some studies being going on in plant accumulation, but I'm not 100% sure if the study has been done particularly in this area. So that I'm not 100% sure. But there are many studies that will tell you that how toxic metals can be accumulated in plant bodies. There are vast number of studies being done, and some can be found like metals coming out of dye industry also. But I'm not 100% sure if a lot of studies has been done in this particular area, because these are informal dyers, and we had to struggle a lot in order to even get any data from them, because they're very scared of their livelihood. Like if someone goes there and studies them, then they're scared that government might shut down them. So that's why you probably have noticed I've not disclose the location. I just said location A and location B. So yes, but that can be done, but that would need a lot more governmental support. Like we have to be sensitive about these people. But there are some lot of stories in, I mean, lot of studies being done in this in general in this area. Yeah.
Okay. So there is a question from Summer. So what are the some primary environmental stressors or bioindicator that can be present in and inadvertently affecting high density residential urban areas of India?
Uh can you repeat the question? It's a long question.
Yeah. What are the some primary environmental stressors or bioindicator that can be present in and inadvertently affecting high density residential urban areas of India?
I do not remember exactly a name right now in this point, but I would say in if you have to study like urban areas, then lots of macroinvertebrates can become like, for example, oligokit could be an example, and it could be universally and bioindicator for many urban areas, not only in India, many urban areas. Oligokit could be, it's a macroinvertebrate. There could be some bacterial indicators also that could be there, but that's like something needs to be more investigated, and India have to develop more of these bioindicators. I'm not sure if we have a lot of studies, but I know that there has been a lot of studies being done in terms of agricultural bioindicators, because agriculture is a lot of priority in India. So you can look up and see agricultural bioindicators. A lot of plants being used as agricultural bioindicators. But in general, like I said, oligochaetes and several like some diatoms could be some general bioindicators universally present, and that could be applied in India also.
Okay. There is a question from Harsh Yadav. Does it often happen that a particular macro invertebrate significantly vanish from a particular site?
Yep, it can, because of if stress factors increases and it cannot sustain, then it will vanish. For example, bacteria might also vanish because the stress factor is so much, the pollution is so much, then it will totally vanish. It won't be. It will be extinct from that particular ecosystem. Totally can be. They are sensitive species, so they will vanish.
Okay. Looks like there are no more questions. So, on behalf of Envis University of Science, I thank you wholeheartedly for accepting our invitation and sharing your vast knowledge. And also, participants also has asked many questions, and you have answered them very well. So, I hope we will try to extend our webinars in near future, and also we will share some more interesting webinars. So I request all participants to subscribe to the channel so that they will get the updates quite often. So in the end, I would like to thank you once again for sharing your knowledge. Thank you
Thank you
so
so
much.
much. Thanks for having me. Thank you.
Yeah.